Bismillahirrahmanirrahim. Assalamu alaikum, dear all students. Welcome to the new lecture of infectious disease diagnostics. Today our lecture will be about the media used for processing of cultures. In today's outlines, we will be learning about the different types of culture media depending on their uses and depending on the different purposes and samples for the processing, and then we will discuss about the culture media selection, that what type of media should be used for which type of culture and which type of sample. Now our today's first topic, the types of culture media. And culture media is an environment which is required for the growth of bacteria in the lab and for proper identification of any microorganism, especially the bacteria or fungi. This culture is required, because by growing the bacteria and finding out the different properties on the basis of the growth and on the basis of the different strains, these bacteria can be identified and these bacteria can be diagnosed, that what type of infection and which bacteria has caused the infection. If we have obtained the growth and this growth is showing us the colonies of multiple types and the different types of characteristics appear in a same culture plate, then a subcultures or pure culture is required. So the pure culture need a specific medias which have the specific nutrients and specific conditions for isolating a pure culture. Various types of culture media is available. So today we will discuss about the different types of culture media depending on the ability of the growth functions and depending on the support they give to the microbes and bacteria. The first of them is the non-selective media, then the selective media, the third one is differential media, fourth one is enriched media, the fifth one is enriched media or enrichment media or enrichment broth. Then we will discuss the different transport media and the media which is required and used for the identification, and in the last we will discuss broth media. The non-selective media, it is a type of culture media which is required to support the growth of those organisms which are non-fastidious. The non-fastidious are the type of microbes which do not require any specific requirements, any specific media and any specific nutrients and food components for their growth and survival. So that type of microorganisms are called the non-fastidious microorganism. And the example of this non-fastidious, or we can call it a simple media, is a sheep blood agar. This is a standard non-selective media, and mostly in the well developed and standard labs, this is a standard medium used for all the non-fastidious microbes. The second type of media is the selective media. As the name indicates, that this is a type of culture media which support only the growth of one type of bacteria or a group of the one type. So this media is required for providing support to only one type or to one group of microbe. And this media is important because it give favors to go growth for specific microbes, for specific bacteria, and also it also discourages and inhibits the growth of those organisms which are unwanted, or the growth of organisms which is the part of the normal flora. So, selective media is used for selective bacteria or a selective group of bacteria. The example of selective media is the bismuth sulfide agar or BSA. It is used for the culture and isolation of Salmonella species. Alkaline peptone water, this is a selective media which is used for the isolation of gram-negative bacteria, Vibrio cholera. Third type of selective media, that is the potassium tellurite agar, and this media is required for the selective growth and selective isolation of Corynebacterium diphtheriae. TCBS, thiocitrate thiosulfate biosalt agar, it is used for the isolation of Vibrio cholera. TCBS, thiosulfate citrate bile salt agar, it is used for the isolation of different Vibrio cholera species, and Salmonella shigella agar or SS agar, it is used as a selective medium for the isolation of Salmonella and Shigella. Enriched media, this is a type of media which contain growth enhancers, and they enhance, they give support to those organisms which are fastidious organisms, and certain enhancers they are added to that non-selective agar. So, that non-selective agar then becomes a good and enriched media for the fastidious organisms. Fastidious organisms are those organisms which require specific nutrients and food for their growth and culture, and the commonly used substance which are for the enrichment and for the enhance of growth is the blood, the serum, the ascitic fluid, the glucose and plasma. So, the various concentration of these components are required for preparing the enriched media and making the media suitable for the fastidious organism to grow. And the example of this enriched media is chocolate agar. Differential media, it is a type of culture media which allow the grouping of microbes and helps in the differentiation of microbes based on the different characteristics which appears during the growth on the culture medium. For example, certain media they gives color when a specific group or a microbe from a specific group they are cultivated and they show their growth. So, then the colors, different colors or different properties appear. Certain media they are differential and as well as the non-selective. For example, sheep blood agar, this media works and functions as a differential as well as a non-selective media, because this differentiates different organisms on the basis of hemolysis. Certain bacteria they produce beta hemolysis, which is the complete hemolysis of the red blood cells, and certain bacteria they show partial hemolysis or the alpha hemolysis, and certain bacteria they show no hemolysis and the gamma hemolysis. So, The media, differential media, can also be become selective. For example, McConaughey agar. This agar works as a differential as well as a selective media in certain circumstances, because this McConaughey agar inhibits gram positive bacteria and also differentiates gram negative bacteria on the basis of lactose fermentation. Certain gram negative bacteria, they give pinkish color when they ferment lactose in the media, and those bacteria which do not ferment the lactose, they do not produce that pinkish color. Clad medium is another example of the differential as well as the selective medium. Clad stands for the cysteine lactose electrolyte deficient medium. Enrichment medium. This is a type of medium, usually a liquid, which enhances the growth of small numbers of specific organisms and suppress the other normal flora. So, these organisms are usually liquid in their structure, liquid in form. So, enrichment broths, they are incubated for a certain period and then subcultured to isolate the specific species from that broth. The example of this enrichment media is the tetrathionate broth and the selenite F broth. The Salmonella and Shigella species are enriched in the selenite F broth or the tetrathionate F broth, whereas the other intestinal flora, especially the E. coli, Klebsiella, these microorganisms are inhibited by this selenite F broth. And sometimes the environment also act as an enrichment media, and it is also modified for supporting certain type of microbes and inhibiting the other. And cold enrichment, it is used for the Listeria monocytogenes, whereas heat enrichment, it is used for Legionella species. Because these microbes they grow well under these conditions, and other microbes are the normal biota, they are inhibited. The Listeria monocytogenes, it can grow on the lower temperature, while most microbes they grow at the room or at the body temperature, whereas the Legionella species they grow on slightly higher temperature, while other microbes they cannot survive in that higher temperatures. So, that why this temperature can also be used as an enrichment media. Transport media, it is a type of media, it can be semi-solid or solid media, as the name indicates, used for the transportation of the different samples to the microbiology labs, and it contains ingredients which promote the survival of aerobic as well as the anaerobic pathogens. And these transport media, they prevents the growth of commensals or the normal flora, and required when specimen is to be shipped or transported from the lab where this culture facility is not available, or if sample is sending for the reference lab. The example of this transport media is the Kerry-Blair medium, and this is used for the overall enteric pathogens, whereas Amis transport medium, it is used for the viability and survival of gonococci. So, these are the two examples of transport media. The identification media, it is required for the identification of certain bacteria, in which when substrate or certain chemicals are added, So after addition of these substrates or chemicals, the certain changes occur in this media. On the basis of their changes, we can identify different bacteria. The example is the Bactone water, urease agar, or Kliegler iron agar, or simply we can call it triple sugar iron. These identification media changes their color, or they produce gas, or they give any specific kind of chemical substance, they produce any specific kind of chemical substance which is helpful in the identification of different bacteria. Broth media is a type of supplemental media which is used to aid to different agar plates to detect minute number of the microbes that can be aerobes, anaerobes, or microaerophils. And the example of this broth media is the thioglycolate broth, and this media is mostly used as a supplemental media for the identification and detection of different microbes. The next topic of today's lecture is the culture media selection. On the basis of the different specimen submitted in the lab for culture purposes and the availability of the organisms or the suspicion of the organism that what kind of microbe is suspected in this specific specimen, different culture media are selected. The samples in which the different fastidious pathogens are suspected, then a specific nutrients or enhancers must be added to the media to support in their recovery, so that these sample may not goes in vain. So therefore these fastidious pathogens, they require a specific nutrients, and that nutrients should be added to the media. If a sample is collected from that site which have normal flora, so that sample require a specific selective medium, because that selective medium that allow the pathogens and that also depress and decrease and inhibit the growth of the normal flora. The primary culture media, it is selected on the basis of the past experience, the patient population, and any special circumstances. Primary culture media is that type of culture media which is required for the initial inoculation of the sample. For example, if sample comes to the microbiology lab, so depending on the type of sample, which type of media is used, or simply in the routine labs differential media is used, so that is called a primary culture. And that culture media selection, it is based on the past experience, that if we have the lab have tested different media, so on the basis of that testing, what kind of media is giving good results and producing more bacteria and the correct type of microbes and pathogen, so the lab will use that specific culture media. And if there is population depending on the incoming number of samples, so if the sample number is high, so then the basic or the specific media can be used, because then the subculturing or reculturing of the media that of the sample, that will need more time, and timely reports cannot be generated. So therefore the patient population also affects the type of culture media. And if there is any special circumstances, and if there is any special media requested by the doctors, So that media can be used on the basis of their requirements and their circumstances. In some conditions, a series of different plates, multiple plates, it is required. These plates, this culture media, they are labeled and arranged in a specific order. For example, if we are testing for the new kind of microbe, so we will inoculate that microbes on various plates, and their arrangement order will be beginning from the most enriched medium, and then their enrichment decreases and going towards the most selective medium. So this type of setting is performed for any new specimen or any special requirement. The culture media selection, it include the different terms and the different items for the routine culturing. The first one of them is the non-selective agar, that basically when sample comes to the labs and that is received, and during inoculation it is primarily inoculated on non-selective media. And then if fastidious organisms are suspected, or the sample is from the sterile body fluids or sterile body sites, so enriched media is required for that kind of sample. The third item, the third type of media which can be selected, selective and differential medium. So if the sample is from the enteric tract and gram negative bacilli is suspected, so for the most routine cultures, selective and differential medium is required. For example, McConkey agar. So McConkey agar can be used as a selective and differential medium if the sample is from the enteric tract. Selective medium, it is used for those organisms which are usually gram positive, or the specimens in which different mixed gram positive microbes are suspected, and both gram positive or gram negative bacteria is suspected, then selective medium is used. Additional selective media or enrichment broths, it is required for specific pathogens if needed. Broth medium, it may be used as a supplement with specimen from the different sterile body sites, or from where minimum amount of bacteria are suspected and their numbers are minimum, or if there is a tissue collected and submitted in the lab for the processing, or the lesions, the wounds or abscess. So broth medium is added to these type of specimen when they are collected in the microbiology lab. Now let us have a look at this chart, which shows us the different types of specimen and the selection of culture media for different specimen and direct gram stain. So this is the list which shows the different types of specimen, and the direct gram stain is either performed or not. This is a blood sheep agar and the chocolate agar, McConkey or EMB, eosine methylene blue agar, the anaerobic culture thioglycolate broth and Thier-Martin medium in case of certain specimens, and if any other type of culture media is required for this specimen, then they are listed here. So the first of these specimen is the aqueous or the vitreous blood. If blood is collected and they are received at microbiology lab, so the gram stain is performed for this type of sample, and then they are inoculated on blood agar as well as chocolate agar for the fastidious organisms. And then in case of differential and selective medium, then McConkey or EMB agar can also be used. They are usually not cultured anaerobically, and the thioglycolate broth for their support is also added to them. They may be cultured in blood culture bottles. The second type of specimen, which is the body fluids, they contain the amniotic fluid, the bile, bone marrow, pericardial, peritoneal, and the fluids from the different areas and species, the viscera of the body. So, if amniotic fluid is selected, then it is inoculated on all the plates, and if sufficient volume of fluid is present, and if a sufficient volume is not present, then the gram stain and blood agar and anaerobic culture is performed for this type of specimens. If bile fluid is received, then their gram stain is performed along with the blood agar, sheep blood agar. They are not cultured on the chocolate agar because the fastidious organisms are not present in bile, and there is a chance of gram negative aerobes. So, they are cultured on McConkey or EMB. They are not cultured anaerobically, and this thioglycolate broth can be added to this. Bone marrow is also stained with the gram stain, then the inoculated on blood and chocolate agar because these may contain the fastidious organisms. Pericardial and peritoneal fluid is almost they are processed in the same way, and both have same processing, but the pericardial fluid, it is sometimes cultured anaerobically because it may contain the anaerobes, and the pleural and synovial fluid, it is processed in the same way. Both are cultured on blood sheep agar and chocolate agar because these may contain fastidious organisms. And the peritoneal fluid, it may also be inoculated on Thier-Martin agar because the some cocci which causes the inflammation of the brain and the layers of the brain. So, that's why meningococci, they are inoculated on this Thier-Martin agar. The catheter tips, they are inoculated on the blood agar because it may contain various types of microbes, and then after this primary inoculation, they are then further processed for the specific or selective medium. CSF, it is gram stain initially, then it is inoculated on blood and chocolate agar because there are chances of this fastidious organisms, and it is gram stained, but before gram staining, centrifugation is important, and it is recommended for the gram staining of CSF. Ear samples, both the inner and the outer ear samples are processed in the same way, but to the outer ear, thioglycolate broth is not recommended because it contains the normal flora. Eye specimen is also processed, and all the media can be used for the inoculation of eyes specimen except the anaerobes because anaerobes are usually not present in the eye infection. The gastrointestinal samples, they are processed if a duodenal aspirate or gastric aspirate is received, then the aspirate are processed both and they are inoculated in the blood sheep agar and chocolate agar along with the McConkey or EMB agar. If other medias are available, for example CNA, then the duodenal aspirate is also inoculated on this CNA agar. CNA agar is the update of another form, updated form of Columbia agar in which nalidixic acid is added before the processing. So, that is why It is also used for duodenal aspirates, that it is become selective for the specimens. Genital specimens, the vaginal or cervix samples, they are almost processed in the same way, and all of them are gram strand, and then they are inoculated on blood and chocolate agar. But the vaginal samples or cervix samples, they are inoculated on McConkie agar, and both the samples are inoculated on Thier-Marter agar, and to check the gonococci if present in them. If genital screening is performed for Neisseria gonorrhea or group B streptococci, then if Neisseria gonorrhea is suspected, then they are inoculated on chocolate agar and Thier-Marter agar, and if the group B streptococci is suspected, then they are inoculated on blood agar. If the sample is collected from wound, abscess or any lesion, then they are inoculated on all of the media present, because it may contain the aerobes and as well as anaerobes and the fastidious organisms. If there is a sample from the lower respiratory tract, if there is bronchial washing, bronchial brushing or lavage, or if there is sputum, then they are inoculated on the blood agar, chocolate as well as EMB or McConkie agar. They are not inoculated on anaerobic media. But if the sample is from the upper respiratory tract, then usually their gram stain is not performed, but they are inoculated on the blood agar initially. And the suspected fastidious organisms in nasopharynx are sinus aspirate, so then these are inoculated on the chocolate agar, and sinus aspirate is also inoculated on the McConkie agar. If there is a tissue specimen, then tissue is inoculated on all of the agars, blood agar, chocolate agar and EMB agar, and it is also processed anaerobically. And if a urine sample is collected and normal urine sample is collected or catheter urine, then they are inoculated on blood and McConkie agar. And if there is suprapubic aspirate, their gram stain is also performed. They are inoculated on blood agar, McConkie agar, anaerobic medium, and thioglycolate broth is added to this suprapubic aspirate, because there are minimum chances of the normal flora in suprapubic aspirate. That was all for the topic media used for the processing of cultures. Thank you for watching. If you have any question, you can ask. Assalamu alaikum. Allah.